Hi friends. Today I wanted to discuss the revision capsule of meiosis. Meiosis is also called as reductional cell division. You know that the parent cell undergoes meiosis to form four daughter cells. So this process wherein the parent cell is diploid and the newly formed four daughter cells are haploid. So there is a reduction in ploidy level, because of which we call this as reductional cell division. Meiosis is also called as reductional cell division. So you should remember that meiosis undergoes only one cell cycle and DNA replication takes place only once. And meiosis is divided into two phases, meiosis-I and meiosis-II. So meiosis-I is very important stage, because during this meiosis-I, what you notice is there is a recombination of the genes. Genetic recombination takes place, and you know that genetic recombination leads to variation, and variation is one of the raw materials for evolution along with mutations. So genetic recombination leads to variation and variation to mutation. So meiosis 1 has a elongated prophase stage. So prophase 1 is divided into different stages. You can remember all the stages by the mnemonics LZP D square: leptotin, zygotin, packaging, diplotin, and diakinesis. LZP D square: leptotin, packaging, diplotin, leptotin, packaging, zygotin, diplotin, and diakinesis. So what you have to remember is, in leptotin, the chromosomes are long and slender, and they come together. So in the zygotin stages, the pairing up of homologous similar chromosomes takes place, which we call it as homologous chromosomes. Of this homologous chromosome, one is maternal chromosome, another is from paternal chromosome, from the mother side and father side. So we call it as maternal and paternal chromosomes. They pair together and they are held up. There is a formation of synaptonemal complex. That is in case of the zygotin stage. Pachytene, you find that there is a crossing over of non-sister chromatids, and there is the exchange of genes takes place. Genetic recombination is completed during the pachytene stage. In the zygotin stage, uh, we have already discussed about that. The next one is diplotin stage. Diplotin, what you find is there is dissolution of synaptonemal complex. Whenever there is dissolution of synaptonemal complex, the homologous chromosomes move away from each other except at the point of connection, and they look like egg-shaped structure, which we call it as chiasmata. So chiasmata is noticed. In the last stage, that is the diakinesis, there is terminalization of chiasmata, and there is the degeneration of the nuclear membrane, nucleolus, the disappearance of mitochondria, Golgi complex. All this happens typical to any prophase stages. So prophase 1 is a very important stage in case of meiosis 1. The next is metaphase stage. In metaphase stage, the homologous chromosomes are arranged in the equatorial plate, and only one end of the centromere is attached to the spindle fibers because of this unique arrangement or alignment of homologous chromosomes. Then anaphase 1, we see the separation of homologous chromosomes. There is no separation of sister chromatids, instead there is only separation of homologous chromosomes. So half of the chromosomes move towards one pole, the other half towards the other pole, because of the condensation of spindle fibers. So this separation leads to the haploidy, or reduction in the chromosome number. So in the telophase, there is reappearance of nuclear membrane, nucleolus, mitochondria, Golgi complex, and there might be a cytokinesis, or they might continue without cytokinesis. So telophase, we find two daughter nucleus are formed. See, the meiosis-II, in between meiosis-I and meiosis-II, there is a short interval of resting phase, which we call it as interkinesis. Nothing happens there. So, and I have already told you, only once the cell cycle takes place, that is before meiosis-I. In meiosis-II, why should meiosis-II happen is the sister chromatids are not yet separated. So meiosis-II is also called as haploid mitosis, because the product of meiosis-I is two haploid cells, so dyads are formed. So these haploid cells simultaneously they undergo prophase 2. So there is disappearance of nuclear membrane, nucleolus. The chromosomes are going to condense, and they are forming the chromosome-like structure. And in metaphase stage, you find that the chromosomes are arranged on the equatorial plate. In this metaphase, the spindle fibers are attached at both the ends of the centromere through the kinetochores. So the next step is anaphase 2. So there is a separation of the sister chromatids due to the splitting up of centromere longitudinally. It is similar to the anaphase of mitosis. So the separation of sister chromatids takes place in anaphase 2. In anaphase 1, it was separation of homologous chromosomes. After this, the next stage that we notice is the telophase stage, telophase 2, wherein the daughter nuclei are going to form, and four of the daughter nuclei are formed, and cytokinesis also takes place in this stage. So thus there are formation of four haploid daughter cells. Thank you.